Wat baas nemen.
Ladies and gentlemen, welcome at this academic session in which Professor Harwood will accept the chair Integrative Neuropsychiatric Cell Biology. My name is Stef Kremers. I'm the Vice Dean of the Faculty of Health, Medicine and Life Sciences of Maastricht University, and I'm Pro-Rector at this academic session. It is my pleasure to welcome you to this inaugural lecture. We've come here to celebrate the appointment of Adrian Harwood as professor at Maastricht University. Professor Harwood would probably describe himself as a card-carrying cell biologist. He made his reputation with work at the MRC Laboratory of Molecular Biology in Cambridge, and later as a faculty member at University College London. The expansion of his interests into neurobiology and mental health came through the investigation of the molecular effects of lithium, one of the most effective psychiatric drugs in cellular model systems. After his move to Cardiff University in 2005, he expanded his interest in neuroscience and was involved in the establishment of the Neuroscience and Mental Health Research Institute, now the Neuroscience and Mental Health Innovation Institute, which is a strategic partner of our faculty. However, he is not just involved in cutting-edge laboratory research. Professor Harwood coordinated the cost, action, maximizing impacts of research in neurodevelopmental disorders, which brought together scientists, clinicians, and patient organizations from all over Europe. Such international activities are very close to his heart, which is why he is the perfect person to make academic connections across countries. As a university with a very strong international focus, we are very pleased with the partnership with Cardiff. I learned from his colleagues in Maastricht that Adrian's enthusiasm for science is contagious and that he enjoys contributing his insights to new projects. Adrian is always enthusiastic, has deep molecular knowledge, and he eagerly and effectively shares it with young researchers. Outside work, Professor Harwood is a keen skier and enjoys half marathons in European cities. Sport is a passion that is shared within the whole family. Another passion is traveling, which he does extensively with his wife Janet. And last but not least, he has mastered the craft of brewing and is well known for his homemade special beers. So you know how beer is brewed and probably also why this is so important. Well, this leads me to invite you to the podium. Ladies and gentlemen, I would like to welcome to the podium Professor Adrian Harwood for his inaugural lecture titled How Brains Are Built and Why This is Important.
Okay, thank you. That was more than flattering, I think, actually, and pretty much true. I know who you've been talking to. Okay, so it's actually a great honour for me to take up this position. It's something that fits I guess fits one of my own sort of career highs. And although I've actually become a professor twice before, this is the first time I've ever done an inaugural. So that alone is a pinnacle in my career. So thank you very much. So I'm going to hold this chair, as we just said, in conjunction with my chair in Cardiff University, which I've held for the last 20 years. It's been a great opportunity. It's a great opportunity to really bridge between our two great universities and great institutes. And reflecting on this, I actually realised that I've often done things in doubles throughout my career. And I call that the power of two. So when I first set up my laboratory in UCL, I actually hired 2 research assistants. And then that was followed by 2 PhD students very quickly, and then two postdocs. So I had an instant lab almost. And in our own family life, we had 2 children, we had twins. So we actually had an instant family as well. I guess at this point, Janet might be getting a bit worried about why I'm talking about 2s, but basically the whole point is that if you bring different people together and different people with different ideas and you work together, you actually see things in a different way. And it's a great pleasure, really, to bring together the 2 institutions from Cardiff and Maastricht. So in the double chair, really, I will hold my position both here in the Department of Translational Genomics and also in MENS, Mental Health and Neuroscience Institute. And on the other side, in Cardiff, as we said, I'll continue to be a co-director and chair in the Neuroscience and Mental Health Innovation Institute. And I think together we will be better. So my title is a long title. I gave a quick Google search, showed me that it's possibly a unique title, okay, which is good. You should always strive to be unique and individual. And hopefully in the next 30 minutes, I can explain what it is, why it's important, and actually why you need one now. Okay? And you can tell me later whether you agree or not, and certainly my family will. Right, so, okay, at my core, as you said, I am a cell biologist. I tend to look at the world as a cell biologist. And I did that from my very early days, even as an undergraduate. And the basic idea is that if you can understand cells, you understand the interface between the genetic information, DNA, and actually what it means in the real world. And I would even argue that drugs target cells, not necessarily whole bodies. So I've always studied that. And when I started studying that as an undergraduate, we didn't really have any of the tools that you're completely familiar with now. The way you would do something is to take some cells, put them in a culture, add a drug, see what happens, and then try and work out what happens with that. Now, at that time, we were just getting the development of the molecular genetics, the molecular biology that we're used to today. And in fact, my PhD, I went to Edinburgh and I worked in the first fundamental time when we could control and manipulate organisms through their DNA. And what I did as a PhD student, you would now call genomic engineering, but it just didn't exist at the moment, at that time, genome editing. But we figured out how to do some of these things. And throughout their career, we've applied advancing technologies as they've come along. And really, that gave us the game plan that we would use really to study mental health, brain development, and other aspects of development. And so, for me, the cell biology at the end of my title is really to do with the sort of game plan that we're applying, where we look at DNA, try and understand how that controls the cells and environment around you, and what happens at a cellular level, and then from that, and this is why it's a dotted line, try and extrapolate and think about what that means for human health. Now, also, when I started, we didn't have such things as human stem cells. We didn't have stem cells. Stem cells were invented along and discovered along my career. So we all had to pick simple, easy to manipulate, controllable systems. And I chose to work with probably one of the simplest developmental systems that existed at the time, and probably still does exist, although one could argue about that. And it was not even an animal. It was an organism that was very close to being an animal, called dictyostelium. And the reason we picked that is that, first of all, it was manipulatable. We could work on it in terms of DNA. But also, it had a very simple body plan of 2 or 3 different cell types. Okay. And we worked on that for about 20 years. And we learned a lot. We learned a lot about genes. Many of those genes actually are functional in the brain and really important. In fact, one of the genes has just popped up in a discussion that we've had between Cardiff, even today. So what we learned from dictyostelium, we can extrapolate into the human condition. But 2 things happened. Okay? The first thing happened is that although our game plan was really to manipulate DNA and then think about the consequences of that, what actually we thought eventually, that wouldn't it be really clever to combine drugs and DNA manipulation? And we had this idea that we'd be able to then find more and more and to delve deeper and deeper into developmental biology. And we happened to pick a drug that was very easy to apply. There was many reasons why we wanted to look at this drug, but also we knew it had a developmental impact, and that drug was lithium. And it turned out to be almost a career-changing event because it opened up a whole area of trying to understand how psychotropic drugs work. And we learned a lot about lithium in that time. But the one thing that really surprised me is that we thought it would be so easy to just look up in the medical textbooks why people use lithium for bipolar mood disorder, which is still its most common use. Okay? And we thought we'd just look it up and it will tell us the answer, and then we can understand developmental biology. And we were fairly shocked to find that no one knew. And in fact, even now, even though we've learned a lot about lithium as a drug, I think we still don't know how it works in mental health. So there is an unfinished bit of business out there that maybe before the end of my career we'll work it out. But that got us interested in mental health. And in fact, we wanted to really understand and apply what we knew to understanding mental health and using and designing better drugs. A new lithium, or a new antipsychotic drug. And so that got us into the middle part of my title, which is neuropsychiatric. And what we mean by neuropsychiatric neuropsychiatric conditions is really that combination of neurodevelopmental conditions, psychiatric conditions, such as schizophrenia, ADHD, autism, and actually intellectual disabilities, that affect how the brain develops and affects earlier stages of life. And the neurodegenerative neurological conditions, such as Alzheimer's and other dementias that affect the end of that life. But we're looking at it from the perspective of cell biology. So we are thinking about what is the cell biology that's underlying these processes. And actually, it's quite similar. It may be different because of different parts of your life. It may be different the way it manifests itself, but you have neurological conditions that affect children and you through your lifestyle, lifespan, including epilepsy, and also our brains continue to develop, even now when you become a professor at whatever age. It's a constant process that's going on at the cellular level, and there's a lot to learn in combination with that. So why is this important? Well, certainly when we look at the conditions, both neurological conditions and the neuropsychiatric and the psychiatric conditions, we're talking about 2 of the top 10 chronic disabilities and illnesses in the world today. So it's a major global issue. And if you pile them all together in every single possible way, the sort of thing you might do if you were writing a big grant, for instance, over a lifetime, 30% of the population are affected by one of these conditions. Okay. So it's affecting 30% of the population. So it's a lot of people if you think of the global population. So it has a high socioeconomic cost. It costs health. It costs work, family and carers, society in general, because it has an impact, and even work productivity. So its impact, or the impact of mental health issues, are massive in the world. Okay? And yet, it's incurable, and the drugs that are currently available target the symptoms when they are available, and not really targeting the underlying causes. And so there's a huge need for drugs in this area, but actually there's very little activity because it's so difficult. So if you like, our role is to try and lower that energy barrier to delivering better drugs. Okay. So at this point, I would actually like to point out that by chance, but also by fortune, this is Brain Awareness Week. So it's actually a pleasure to give this inaugural lecture during a time when actually we should be thinking about the brain. And I will give you my tiny, tiny contribution to the drive to make people really understand how important this is and actually get progress being made. So, and that brings me to really the last part, or the first part of my title, which is integrative. Right? So we've got cell biology, that's what I do, that's my core skill. Neuropsychiatry, which is in psychiatry, which is the really important aspect of what we're doing now. But we need to integrate it, okay? So what does that actually mean? Right, so as I said, my game plan, or our game plan, the game plan everybody's ever come in contact with possibly, is to really think about how the genetics of mental health affects affects its effect, it delivers its effect on the individual. And the second most important event, or the second important event in my career, was actually the discovery of stem cells and the technology development that's happened in the last 10, maybe 15 years, but 10 years probably, to really enable us to take cells from patient blood and turn them in, take blood from cells from patient blood, and reprogram them into stem cells, and that we can then differentiate into neurons, and effectively we can now do human cell biology, or an aspect of that, in the laboratory. So that is effectively taking that very simple system that we had to the other extreme, to probably one of the most complicated systems that you could think of as a developmental biologist. The development of the brain is the expansion from one cell, or a small group of cells, all the way to billions of cells in our brain, which then connect together to form trillions of connections. So it's probably one of the most complex organisms you could possibly, organs that you could possibly think of. And so we want to know how and why. Now, in principle, genetics will lead us the way. In practice, there's a few complications. Right? And the first complication is, we'll move on. The first complication is that genetics of mental health is incredibly complex. For most of us, we carry some sort of level of risk of mental health issue. But there is no single causative gene for any of the diseases. There's a few special cases, but whether somebody develops a condition or not is actually a combination of lots of genetic factors. So in terms of the simple idea of manipulating DNA to understand this, I mean, we can't do it. It's just too impossible to sort of model a human being in a cell in mental health research. Okay. But there are some ways around this, and I'll come back to that just a bit later. The second problem is there's lots of things going on. There's not one single mechanism that causes, for instance, schizophrenia or autism. There's lots of things going on which are all being affected by the genetics. So we've got lots of pathways and complex genetics. And as many of the people here in the audience know, the actual outcomes for an individual, for a patient, is incredibly variable and complex as well. So we've probably picked the hardest genetic system that you could possibly imagine and the hardest developmental system you could possibly imagine to work on. But we all like a challenge, and it's an important challenge. One of the ways to tackle this is actually just, if you like, the most direct way. And that's really to think about, well, why don't we just get lots and lots and lots and lots of patient stem cells and just study cell biology of the patient population? And that is a major enterprise. It is a huge amount of effort. But actually, we've set up a spin-out company to start that process. So we already have a company that's taking the direct line. But our role as academics, I think, is probably to think about faster, better, other ways of doing this that would enable us to deliver, or at least understand enough of this process to be able to deal with it in a more approachable way and to deal with it in a faster way, and in order to be able to just understand a lot more of what we need to do and ultimately make better drugs. So I'm going to take a slight diversion here, really to try and illustrate how we're thinking, or how I'm thinking about this as an academic, and how my group is thinking about this as an academic. So I want you to reimagine mental health, okay? I want you to reimagine mental health. Mental health usually means some condition that affects patients. I want you to reimagine mental health as a healthy brain, right? Okay? And if you think about it, I gave you that figure of 30% of the population at some point in their life being affected by some sort of brain disorder or some brain-related condition. Okay? But 70% are not, right? It may be that it's sub-threshold, it may be it's fine, but it's not. 70% of the population don't really require any clinical intervention, don't really require that sort of support. We're all different, and we all have different days, and good days and bad days. But if you think about it, you could argue that the brain is actually really, really good at being healthy. You know, that's probably what it's really clever at, is staying healthy throughout our lives. You know, when we're tired, when we're stressed, when we get old. You know, and there's when we have to eat terrible diets, or maybe when we've been out enjoying ourselves. So the brain is really good at actually staying healthy. And you could think about this in a different way. You could think, you know, the brain works in operational parameters. You know, it can deal with a lot of things, which is this blue halo I drew around this image. And if you stay inside those operational parameters, you'll be fine, right? But from time to time, in individuals, or time to time in all of us, you drift outside those, and that leads to a sort of lack of health, if you like. So you could sort of try and reimagine brain and mental health as a sort of loss of good health rather than gaining bad health. Okay. Now, you'd think that could be just a sort of semantic English argument about why, just looking at things the other way around. But I would argue that it actually helps explain some things. And it helps to explain some of this complexity. So although there's a genetic component that's inherited for many of these disorders, to a greater or lesser extent. It's not the only thing that leads to having a mental health problem. And in fact, it's always been known that it's a combination of environment and maybe genetic predisposition. Okay? But what's been really challenging is that when you think about all of the environmental conditions that may lead to poor mental health, and you think about all the genetics that may lead, and all the pathways that have been identified that may lead to poor mental health, there's so many of them, and then they overlap. One condition has this similar or even or the same genetic risk as the environmental risk. So if you try and identify one single gene, one single pathway, one single outcome, you're probably missing the point, which is that it's a combination of risk factors that build up. And if you understand that, then, of course, it completely changes the way you might think about preventative medicine and way you might think about dealing with patients, or how you might live your life, right, if you knew where you lay in this spectrum of risk. Okay. So I think that actually, although it sounds simple, is actually probably quite an important point to keep an eye on. So I'm going to illustrate it. I'm going to illustrate how we might work in this area with an analogy, okay? And I've chosen this analogy because I think it will speak to everybody, right? So think of a house. Don't think of a brain. Think of a house, okay? Now, hopefully you're thinking of your dream house. Maybe you're not. Maybe not. Maybe you're thinking of the horrors of your house. But think of a house. But I can guarantee that you'll all be thinking of different houses. Houses are all different shapes and sizes. I don't think anybody's house is the same as anybody else's. It might be a different size. It might be blue. It might be white. It'll have different furniture inside it. You might have got different numbers of rooms. You may have put different books in there. You might have different utilities coming into your house. We probably all have water coming into there. We probably all have electricity. Maybe some of us have gas. Maybe some of us shouldn't have gas. Right? Maybe many of us have an internet connection. Maybe you could take it as far as, we have streaming, right? And we get to choose what we see through our internet connection. I mean, Paul and I, as you alluded to, sports channels definitely, but other people maybe want other things, right? So we all have houses, or we all know about houses, but we will consider and think about them differently. And that's because we probably have different needs and different ways of seeing the world, okay? But if I draw that, which is actually a line drawing, you will instantly recognize as a house, right? It's got all the features in common of a house. It's got walls, it's got a roof, it's got a door, it's got windows, And you think about it, we all know what a house is, and it all has a basic function, and it's a common function, right? But all our houses are different. Yep. Now, anybody who's had a house, or anybody who's thinking about buying a house, will know that actually the most important thing is to keep your house, keep its integrity. Make sure it's got all the things it really needs. Make sure that you, first of all, build a good house, and secondly, make sure you maintain a good house. Okay? And it doesn't matter whether your house is the same or different from anybody else's. The basic thing is, it needs to function as a house, right? Okay? And so we all know that's really, really challenging to do. Right? It's expensive, it's hard work, and houses always fall apart, right? Okay, and need constant maintenance. Now imagine a house where you didn't have to really build it yourself, and you didn't really have to maintain it. Imagine a smart house, one that built itself, and it repairs itself all the time, and it leaves you to do what's the important things like, football and science programs and things like that. It lets you just do the functional, executive things. It runs everything else itself, right? Okay? Now, you could argue that sounds a bit like a brain, right? Okay? So, a smart brain house. Now, there's one catch with our smart brain houses, that we have amnesia. We woke up in this fantastic smart brain house that does it all most of the time. We haven't got an instruction manual. We don't know how it's built. And that actually impinges on our ability to sort of keep our brain healthy, if you like, and how to deal and treat conditions and how to maintain conditions. So, in this analogy, we we basically have this fantastic house that's associated with us, but we have no idea what it's doing. Now, people like me, and actually many of you in the audience, what we're trying to do is actually figure out how it works, how it was built, what it is. We're trying to understand the basic mechanisms and ideas and principles that hold these houses together. Okay. So, I'm going to tell you about one set of experiments, and I'm going to tell you at a very high level that we've been doing in my laboratory in Cardiff, and I think, as many of you might know, we want to do it here in Maastricht as well for various reasons. And it's one condition that we've started working on, okay? And the condition is one of those genetic cases where the risk and the gene correlation with the change is very high, okay? And it's a syndrome called Kleefstra syndrome. And actually, our colleagues in Nijmegen and Erasmus universities now have always been working on this, but it's actually a fascinating condition. It has a number of outcomes, and those outcomes include intellectual disabilities, so cognitive abilities, and actually a risk for psychosis. And we got interested in this because the same gene that generates it, so it's very rare, fortunately, as a condition, but it's caused effectively by one gene, okay? And that gene came up in the bigger patient genetic studies as well. And that gene, and it doesn't really matter what it's called EHMT1, and it's a gene that regulates DNA. Okay. So, what was really interesting about this condition is that if you take stem cells from patients and you then start to develop them, and when we do it, we develop them into progenitor cells first, and then eventually into nerve cells, called neurons. If you take those patient cells from closer patient cells, they actually develop faster. So they form neurons faster than from everybody else, really, from the controls. So it's got a very strange sort of weird, you imagine that something like a mental health issue is a loss of something, but actually we've gained a cellular property here. It moves faster. Okay, so why? And that's the question that really we need to be asking. So why does this happen? Well, what you may all know, but maybe some of you don't, is that the genetic information that makes our cells, makes our bodies, is really encoded in DNA, but makes a copy of something called RNA. Okay? And that goes on to make proteins. It's what they call the central dogma. We've known it for 70 years, and basically it's a linear flow. But what you might not know is what the proteins do, okay? And that's the really key question. So what happens in Kleefstra syndrome is that the proteins that are targeted are proteins that stop neurodevelopment. So if you like, what you've done is lost, or what will happen is you lose the regulators that stop you making neurons, so you make them faster. And in fact, it turns out that it does it in a quite complicated way, in that it doesn't do it directly. It does it through a small RNA molecules called microRNA, which turn out to be incredibly important in our bodies, and a subset are changed in Kleefstra syndrome. And these microRNAs, what they do is actually inhibit the RNA. So they basically inhibit the RNA, that stops the regulators being formed, and then enables cells to become neurons. And what happens in Kleefstra's, and also just to make life complicated, the regulators also regulate the microRNA. So effectively, you've got a brake on development that's actually self-regulating. And what happens in Kleefstra syndrome is that the microRNAs increase. So microRNAs go up, and neurodevelopment occurs faster. Right, so if you're following this argument, you're thinking, well, isn't the important part of our brains having neurons in there, having nerve cells in there? Why having more having nerve cells faster, why is this a problem? Surely it's better. Okay. And in fact, in the laboratory, we now can make nerve cells, neurons, in 3 days if we get the conditions right. So we can make a neuron anytime. And these neurons are perfectly good. They're as good as a neuron that we would have spent 100 days making it through the method that you can see here. And so why is this a problem? Well, we believe that it's not about being a cell. It's not about being a nerve cell. It's about when you become a nerve cell and where you become a nerve cell. So this is not about generating cell types. It's about positioning and placing them. Okay. So why is that a problem? Okay. Well, in the brain, it's well known that microRNAs regulate these sort of processes. Many of the microRNAs that have been looked at in the brain actually have genetic inferences that suggest that they're even involved in mental health. So we know that they're important for the brain, but what we didn't really know is why they're important. So I'm going to give you my house analogy again. So if you're building a house, if you just left it to the builders and with a big pile of material to build a house, I would guarantee that that house is not necessarily as strong as if you had a project manager or a site foreman who made sure everything was ordered on time, in the right place, got it all organised, and directed when the house was built. So when, put the walls on, build the walls before you put the roof on, put the door in after you built the walls. That sort of thing, right? And I would argue that what we're looking at here are the sort of site managers and the controllers of the brain being built. And I'm going to tell you in another way. I'm going to tell you the same thing, completely the opposite. So I think most people are aware of the child nursery tale of the three little pigs. And the basic, I finally after 60 years, we figured out what that was about. So if you think about it, three little pigs, it's about three little pigs who build houses, right? And the first little pig builds a house out of straw. It's got straw, it's the quickest thing to do. Nice build, house. The second little pig considers it a bit and then builds a house of wood, gets a house of wood. Third little pig actually puts a huge amount of effort into building a brick house, okay? Now, at the end of the day, they're all living in houses. They're all quite happy. There's no issue at all until they have an adverse condition, right, in the environment. And in this case, in this paradigm, in this story, that adverse condition is the big bad wolf, but maybe for our brains, it's a few other things that are a little more subtle, okay? And so what we're saying is that everybody can, everyone can build a house, but different houses have different strengths and different robustnesses, and they can resist other things in the environment. And so what we think is happening in Closter syndrome and other syndromes, including 22q11 deletion syndrome, where we know that there's misregulated microRNA, we're thinking that what's actually happening is, we haven't got the organisation right to build a robust, strong house. Okay. So I think that's important. And what I've really told you about is one aspect of this healthy brain idea. So I've told you about the neurodevelopmental aspect, but what you've got is all these factors that are all impinging on the structure and the threat and the risk to a brain. Okay? And it probably doesn't matter which ones they are, because it's to do with how they come together. Right. So I'm going to move this analogy on into the question that's now interesting us now. Right. So what we've told you is that we believe, and hopefully I can convince you, if not now, in the future, that how you put the brain together is really important, okay? But what we actually want to know is, when does that occur? Because if you knew when it occurred, then you might be able to think about how you would avoid these issues. Okay? And so I'm going to tell you about what happens next. And basically what happens next is that you make nerve cells. But you don't just make one type of nerve cell in your brain. There's actually many types of nerve cells. And I've drawn the three that probably interest us most at the moment in the brain. Okay? And these nerve cells effectively are the electrical wiring of our brain house. Okay? And so we have neurons that get excited, and they're driven and make a molecule called glutamate. We have nerve cells that inhibit, and they're often driven by the glutamate neurons, and they make GABA. So in a sense, you've got nerve cells that switch cells on, and then and those cells switch them off. Okay? If you break that circuit, then you've got overactivity. We also have nerve cells that change the sensitivity of the excitatory neurons, and they make dopamine. So we've got at least 3 cell types that we need to think about in these conditions. But there's actually more as well, so we can make this more elaborate if we want. But these are the 3 that concern us. Genetics and the population, although it's not telling us specifically what happens, it's actually pointing to GABA and glutamate signaling being something to do and something important in terms of the risk for schizophrenia, for instance. And we already know that overproduction of dopamine is also seen in schizophrenia patients. And we actually know that antipsychotic drugs target that signaling. So these are not just picked out at random because they happen to match the colours of these wires. I picked them out because these are probably part of the wiring and circuitry of our brain. But the question we really have is, when do they occur? Does that come up? Yeah. When do they occur? And so what I've told you is that the organisation of the brain is about delivering the right things at the right time, to get that right connections in your brain. But you need to get yourself organised before you do that. And so the question we have in our mind now is that occurring a lot earlier than we think? Is it occurring in the very early stages of brain development? And it is known that, in the adult, you still get nerve development, so this could be persisting throughout our life. But certainly in the developing brain, when does this occur? And so the question we've set ourselves now, and actually my colleagues here helping us answer, and in Cardiff, is when does this happen? And our guess from the microRNA experiments is that would happen before we see any neurons, and in this stage that we call neural progenitor cells. Okay? But the problem is we can't see it, because all those cells look exactly the same. And so we need some way of coming into it. So actually when the university opened this year, we had the Einstein telescope given as a presentation. It's a telescope that's not like any telescope we've seen before, one that's based on gravitational waves. It will let us look at the cosmos in a different way and look at time. So I'm going to introduce you to what I think is our cell biology 21st century microscope. And you're all aware of it in the audience, or many people are. And basically, when in the 19th century, a microscope looked like something in a box that you took out and you looked at cells down. And as Janet knows, we've already got one in our attic, actually, because I love old microscopes. But actually, our 21st century microscope looks like a series of boxes. And what this microscope is it's an approach and a technique to look at the RNA on a cell-by-cell basis. So you can look right into the changes inside the cell. Okay? And so you can take some cells. You can sequence all the RNA molecules in all of the cells, and then you can reassemble them and use that to see the cells, right? And you can plot that any way you want. And in this corner is a plot that's really plotting cells, clustering them together by how similar they are to each other, and actually you can then do that over time. So we can now look deep into cell biology and developmental biology, and we can look at the timing, and we can look at all the different dimensions that are occurring, so time and relative dimensions. And it's really about how you put cells in the right place, ready to turn into nerve cells. So we worked really hard actually to try and get this piece of data here for this talk, okay? And this actually is a collaboration that's gone on between Cardiff and Maastricht. It's a collaboration where we had developed the cells in my lab in Cardiff, and we did the sequencing here. And then Janet, who's sitting in the audience, did a lot of the hard sweat to actually do the analysis, especially in time for today. But it actually tells us a lot. So this are the cells where I put that question mark. So this is a time and space plot, identifying how those cells that apparently are doing nothing are changing over time. Okay, right, let's see if I can get on here. Okay, oh, we can see it's moving. Right, okay, so day 0, we have these black cells. So what we've done is superimposed time here. So day 0, those are the stem cells. They're not neurons still, they're just stem cells. But within about 3 days, you get the generation of recognizable neural progenitor cells. So they start to become progenitors almost simultaneously, very, very quickly. And then actually what happens is that from there, we start to see the emergence of cells that will become neurons. Okay? And those cells actually go on to make glutamatergic neurons. But actually what also happens is that the neural progenitor cells then change and they express another gene. Okay? And they express lots of other genes, but they express a gene that we've picked out here called FOXG1. And those cells then go on to make the GABA cells, generating cells, and the dopamine cells. So although we're looking at neurons at the end of the day that are different, they actually started off and sort and organize themselves at different times in different places. And actually, in this experiment that we've shown you, or this interpretation, the change in cell type is actually driven by FOXG1. And what's really interesting about FOXG1 is it itself is a neurodevelopmental disorder. And if you are unfortunate enough to have a mutation in FOXG1, you develop intellectual disabilities. Okay? And you can see here it working in practice. Okay. So we want to take this one step further, right? This is leading really to the heart of why we need integrative neuropsychiatric cell biology. So not only can we see the cells based on their RNA profile, if we know things about those RNAs, we can then sort of paint these cells in different ways. So if, for instance, if we know that an RNA molecule that we're looking at is differentially expressed, a change between a patient and a non-patient, you can ask, where is that happening? Okay? If we actually knew how the symptoms, and can find how the symptoms relate to particular profiles, we could even ask, when do the symptoms or the effects appear in development? So we can start to paint this any way we want. Okay? And in fact, that gives us a route out of here, because, let's just go back one slide. If you think about it, the DNA sequence is something, or the sequence is something, a piece of data that's actually very much in common from one experiment to the next. It's a portable, machine-readable language. It's incredibly malleable in terms of how we can do analysis. And of course, you can do lots of things with that. So you could use the cell biology to ask the questions about, well, which of those original mutations are really important in terms of the experiment I'm looking at? You can ask about, where do these genes appear in the real brain, which is called spatial biology, or brain atlases. You can look at expression of some of these RNAs in blood and ask the question, what am I seeing? Am I seeing this developmental change even in a patient as I look at them? And of course, you can look at different patients and ask, why are people different? And so I would argue that we've got a way now of connecting on many of these quite different things together. And that is my vision, or our vision, I think, of integrative neuropsychiatric cell biology. So just to summarize the science, and I'll just thank everybody in a minute after this. So basically, what have we found? So we found, I think, by looking at the healthy brain, rather than specific conditions, we'll ultimately be able to make sort of predictive biological models. So we'll be able to make ways of trying to predict what happens. Okay? And that will be individualized, so it might tell us about the individual. It will enable us to tailor therapies, and it would enable us to deliver what you call precision psychiatry. And I would argue, and I have argued just now, is that understanding the cell biology of this is really a key instrument in making this become real. Okay, so that's a bit of science, but I just want to also thank where I came from, okay? And I picked this saying, it's a very well-known saying, if I have seen a little further, it is by standing on the shoulders of giants. And it was a quote from Newton, okay, gravitation again, okay, to Hooke. And Hooke was the individual who named the cell. He's the first person who looked down a microscope and said, you know, these look like cells. And he actually meant prison cells, but we now use that term in a different way. And he was writing to him, and he was basically saying, I couldn't have done any of this without everybody who came before. Okay. So who came before? Right. In chronological order, I had a PhD supervisor a long time ago called Chris Bostock, and he basically taught me how to do science. He was a great man. And when I became a first postdoc, which was at the ICRF, the CRU UK, I had a great boss, I guess, Jeff Williams, and he taught me how to deliver great science. And there's a subtle difference between doing great science and delivering it. Okay? And then when I moved to Cambridge, Rob Kay taught me how to actually go for the really big problems. So don't mess around on the margins. Think about what the real problem is and try and answer that. When I moved to Cardiff, it was a change in environment. When I went to UCL, I became a PI, okay, and that gave me the chance to really develop our own thing. And then I went to Cardiff, and we extended onto that, and that moved us in to the whole area of I was recruited by Sir Martin Evans, who discovered, who was part of the Nobel Prize for stem cells. It moved us all into this area of stem cell biology. And it also enabled us to set up the Neuroscience and Mental Health Innovation Institute there. And to thank my colleagues there who particularly were instrumental in that, it was Jeremy Hall, Lawrence Wilkinson, and actually Sir Michael Owen, who taught me everything I ever know about, about psychiatric conditions and mental health. And together we put this thing together. We've worked together in an integrated way all the time. I didn't mention David in that context because David was also one of my colleagues at the time. And he was snaffled away to Maastricht, which I think was probably a pretty damn good move for this university. And knowing when I can see a good thing, he sort of laid the path for me to come here, I think, didn't he? And it took quite a lot of time to do it, but we got here. And so I want to thank David Linden and Theo de Kock, who's my departmental head, who sort of probably understands where I'm coming from, and maybe a bit more now. And we've got together with a fantastic department now, translational genomics, which is exactly the place where one should do this work, I think. And also, I thank Amy Scholes, the Dean, who is my other member of my supervisory committee. I also need to thank all my colleagues that I've interacted with so far. Laurence Denis, Eshen, Pipak, Simona, we're working on, and Julian, Simona Breda, and Julian Kraskoff, Bert Smeets, and Rick, who let us do all this complicated single-cell stuff and made it work. Okay, and I particularly want to thank all the people who have not yet collaborated with me, but you will. And anybody who's spotted me down actually in UNS 50 at the moment will know that my door is open. That is one of my trademarks. I have my door open, and I would like people to come in and talk to me. And sometimes we'll discuss science, and sometimes we'll discuss life. And that is how I try and encourage people around me. And then finally, I should thank Phil and Sue Ambler, who've come here from my oldest college friends, and still our friends, okay, who've come here to see Maastricht, but also recently renovated and built a house. And you were the inspiration for my analogy, okay, along with my son, Paul, who actually works in the planning sector. So in the real world, I wouldn't describe you as a microRNA. You're more a project manager of microRNAs, I think now, aren't you? Yeah. So he also knows why it's so important to manage how you build things. I want to thank my daughter, Claire, who, trapped in her own genetics, has a PhD and is working in the area of life sciences. And she's actually working in a fantastic, great area, which is molecular pharmacology, which is, I think, maybe if I was having my life again, I'd start there. Okay. And finally, I need to thank Janet, my wife, my partner. Yeah, this is real partnership. She's also a damn good scientist as well, which is great, which makes a very interesting breakfast conversations in Maastricht and Cardiff. But she also puts up with me, guides me, stops me going completely crazy, but sometimes lets me do crazy things like, why don't we go and work and live in Maastricht? And we actually love it here. So at that point, I'm going to say, oh, I've got 3 of these, hang on. Oh, no. Thank you, Vel. Thank you. And if we were in Wales, I would also have to say, Joch and Va, right? Okay? And at that point, ich habe gesagt.
Well, Professor Harwood, you couldn't tell that this was his first inaugural lecture, right? Or maybe you could, because all of this youthful enthusiasm coming from this side of the room. Really nice to hear. And with this, I conclude this academic session. We will now go to the reception, and it will take place in the rafter. You will know where it is, because you can simply follow the cortege to the rafter. And my advice is not to stand in line in the hallway, but just to enter the room and then look for a time where you can congratulate Professor Harwood. With this, I would like to close this academic session, and I wish you all a very pleasant day. Thank you.